Hello and welcome to this e-seminar, which will provide you with an introduction to quantitative PCR assay validation. It is assumed that you have a working knowledge of quantitative PCR and have viewed the previous e-seminars on PCR assay design and PCR assay optimization, which are available from the Knowledge Resources section of the website www.gov.uk/government chemist. For those who may benefit from additional information, please refer to the LGC publication Good Practice Guide for the Application of Quantitative PCR, which is free to download from the LGC website at www.lgcgroup.com/NML. In the previous two e-seminars, an introduction to designing quantitative real-time PCR assays and an introduction to the optimization of quantitative PCR assays, guidance and best practice was provided, which covered both the design and performance optimization of in-house generated bespoke real-time PCR assays. This e-seminar will provide you with guidance and best practice on validating quantitative PCR assay performance for commercial and in-house designs, as well as those obtained through the scientific literature. The topics covered in this presentation include how to assess the specificity of a quantitative PCR assay for a DNA target, how to calculate the PCR efficiency and linearity of measurement response, how to determine the working range, limits of detection and quantification, bias and precision of an assay is also discussed. A summary of the topics covered and the recommended acceptance criteria for the parameters evaluated will be provided towards the end of the presentation, along with a list of useful resources and reference documents. Evaluation of an assay prior to use in routine analysis is essential in order to ensure that the results produced in the laboratory are fit for purpose, whether validating an assay developed in-house or verifying an assay adopted from alternative sources. A number of parameters may be examined in order to assess if an assay is fit for purpose. Those highlighted as informative in the scientific literature include those that evaluate assay specificity and those that evaluate assay performance. Recommendations and guidance on best practice in undertaking assay validation and verification are available, but are frequently highly technical and complex in content. This e-seminar will provide an introduction to which parameters to evaluate and how to incorporate them into a stepwise assay validation or verification procedure. For the purposes of this e-seminar, the word validation will be used, but the process is equally applicable to the verification of the performance of assays which have undergone a previous validation. In the following section, a number of approaches for determining assay specificity for a defined target DNA sequence are outlined. As described in the previous e-seminar on qPCR assay design, assay specificity describes the level of sequence match between the assay oligonucleotides, i.e. the primers and probe, and the target DNA sequence. Validation of assay specificity ensures that the oligonucleotides are homologous to the target sequence, do not adopt significant secondary structure or hybridize with one another, and that the reverse complement primer sequence is correctly orientated. To characterize assay specificity, a computer program-based validation, which is sometimes referred to as in silico validation, of the primer and probe designs is performed. The results of this theoretical approach are then normally confirmed through experimental validation. A number of software tools are freely available to assist with the validation process and will be demonstrated on the following slides. The first step in assay target validation is performed in silico using appropriate software. The web-based application BLAST-N may be used to match a primer or probe sequence with potential targets, and can be accessed free of charge at the National Centre for Biotechnology Information website. A screenshot of the graphical user interface as accessed on the 9th of April 2020 is shown on the slide. The dialog boxes for inputting assay primer query DNA sequences and defining the search parameters are indicated. To perform a search, an assay primer query DNA sequence is entered into the sequence dialog box, a job title specified if required, and the reference database to be searched selected. Generally, selection of the standard database option in conjunction with the nucleotide database from the drop-down menu is appropriate. A list of species to search against may also be nominated in order to restrict the search to relevant database entries. In most instances, selecting the BLAST-N search algorithm to identify somewhat similar sequences will be appropriate. Selecting the BLAST-N icon at the bottom of the form will initiate the search, which will be acknowledged on the screen. An example of a BLAST-N search output is shown on the slide. The search parameters employed are summarized at the top of the form, and a match-ranked list of DNA sequences that exhibit significant similarity with the query sequence is given underneath. A series of metrics are provided adjacent to the listed sequences. These may be used to evaluate the significance of the results. Initial match ranking is performed using e-value scores, which give the likelihood that the match has occurred as the result of random chance. The lower an e-value, the less likely the result has occurred by random chance. Hence, BLAST-N sequence match lists are ranked from low to high on e-values and may be used to determine the level of specificity a primer or probe possesses. Target validation should be performed separately for each of the primers as well as the probe where present. It is beyond the scope of this e-seminar to fully describe the match metrics employed by BLAST-N. For further information, you are referred to the reference section of the current presentation, where links to additional sources of information are provided. Following assay target validation, the primers and probe should be evaluated for their likelihood to form secondary structures and hybridize to themselves or to one another. Additionally, this confirms that the reverse primer is in the correct orientation. This may also be performed in silico, and a number of applications are available to assist with the process. Primer BLAST is an example of a web-based application that may be accessed free of charge at the NCBI website. An annotated screenshot of the Primer BLAST graphical interface is shown on the slide. A target DNA sequence is pasted into the sequence dialog box, and the primer sequences entered into the appropriate boxes. Specific primer design parameters may be selected, but in most instances the application may be run using the software default settings. An example of typical Primer BLAST output is presented on the following slide. An example of the typical output for a Primer BLAST primer design evaluation is shown on the slide. The output is described in detail in the e-seminar, An Introduction to Designing Quantitative Real-Time PCR Assays, which is available from the Knowledge Resources section of the website www.gov.uk/governmentchemist. However, in this instance, your attention is directed to the highlighted text boxes. Box 1 indicates the location of the primers in the target sequence. Box 2 lists the sequence characteristics of each primer. Box 3 gives an indication of the likelihood that primers will form secondary structures. Box 4 provides an indication of the likelihood that primers will form primer dimers. Using the information provided from the output of the application, the likely primer and probe performance can be determined. Assay specificity may also be determined directly through examination of the PCR products. This may be performed using either a DNA melt curve analysis or by gel or capillary electrophoresis. With DNA melt curve analysis, the presence of more than one melt peak is indicative of the presence of multiple PCR products, including primer dimers as illustrated here. With electrophoretic analytical approaches, the presence of multiple PCR products is confirmed by the detection of more than one band as shown on the slide. Optimally, an assay should produce a single PCR product, and any other result may be an indication of off-target amplification and hence non-specificity. Assay performance may be evaluated by examining a number of key performance parameters which include the PCR efficiency, linearity of response, working range of the assay, limits of detection and quantification, degree of any measurement bias, and the level of assay precision. In the following section, guidance, best practice and recommendations will be provided for determining the values for each of these parameters. Quantitative PCR assay efficiency may be defined as the number of target molecules present at the end of a PCR cycle, divided by the number at the start of the same PCR cycle. For optimal performance, where the efficiency can be described as being 100%, the number of target molecules should double each cycle. The quantitative PCR assay efficiency may be determined using a standard curve-based analysis. The standard curve may be prepared using a serial dilution of a reference material of known concentration. A 1 in 10 dilution series is frequently employed. It is recommended that the series should encompass a broad range of concentrations in order to span the working range of the assay, with each concentration represented by at least triplicate PCR reactions. As a rule of thumb, a concentration range which spans 5 logs will be sufficient. A graph of CQ plotted against the log of the target concentration should be generated, and the line of best fit determined using linear regression. The PCR efficiency may then be calculated from the gradient of the line of best fit using the equation shown on the slide. The linearity of measurement response may be described by the coefficient of determination or R² value. This may be calculated through standard curve-based analysis as described previously for the determination of assay PCR efficiency. From the line of best fit, the R² value is calculated using linear regression. It is recommended that the R² value is evaluated over 5 independent experimental runs. The working range of an assay describes the range of target concentrations over which the measurement response of the assay is linear. It may be determined using a standard curve approach in conjunction with a broad range of target concentrations. It is recommended to use a dilution series, for example a 10-fold dilution series, spanning a minimum of 5 levels of target concentration and with at least triplicate PCR reaction replicates at each concentration. As a rough guide, concentrations ranging from 50 to 56,000 copies of target sequence per PCR reaction would typically be employed for the evaluation of a taxon-specific marker. Determining the line of best fit to a semi-logarithmic plot of the measured CQ against target concentration will provide an indication of the working range obtainable for an assay. The limit of detection may be defined as the lowest amount or concentration of a target in a sample that can be detected but not necessarily quantified. It may be determined using a standard curve analysis in conjunction with a high level of technical replication. Technical guidance provided by ENGL recommends the use of a level of replication of 60 at each concentration employed to determine the LOD. However, a lower replicate approach may be used in order to obtain an estimate. Two standard curve approaches are routinely employed, and for either option the target concentrations to be analysed should be low. A serial dilution of the target in conjunction with at least a 10-fold level of technical replication at each concentration may be used. Alternatively, a series of independently generated dilutions may be employed in conjunction with a similar level of technical replication. As a rough guide, target concentrations spanning from 100 copies to 1 copy per quantitative PCR reaction should be used. The limit of detection may be estimated as the last concentration at which all 10 replicates return a positive amplification. This should not be less than 3 copies per reaction. It is recommended to perform at least 3 replicate experimental runs. The limit of quantification may be defined as the lowest concentration of target in a sample that can be quantified at an acceptable level of confidence. It may be determined using a serial dilution of the target in conjunction with at least a 10-fold level of technical replication at each of the concentrations employed. Alternatively, a series of independently generated dilutions with a level of technical replication of 10 or more may be used. With either option, it is recommended that the range of target concentrations to be analysed should be low. As a rough guide, a range of target concentrations from 100 copies to a single copy per PCR reaction should be used. The repeatability relative standard deviation should be calculated for each level of target analysed. Guidance on how to calculate the repeatability relative standard deviation is given later in the presentation. The bias of a PCR assay may be defined as the closeness of agreement between the average measurement obtained for a series of test results and a certified reference value. It may be determined by analysing a series of evenly spaced target concentrations which span the working range of the assay. As a rough guide, a minimum of 5 target concentrations should be employed, using a technical level of replication of greater than 3 if practically possible. Precision is defined as the closeness of agreement between measurement results obtained under specified conditions. An example of a metric that may be used to evaluate precision is the repeatability relative standard deviation, or RSDR. This metric may be used to describe levels of within-run or within-batch variation. Repeatability conditions include the use of identical test items, which are analysed in the same laboratory by the same operator using the same equipment over a short interval of time. The evaluation should be performed across the working range of assay, with the metric being calculated at each level of concentration employed for the analysis. As a general rule, it is recommended to use target concentrations that span the working range of the assay and to employ at least 5 evenly distributed concentrations and 15 technical PCR replicates at each concentration. The RSDR is determined at each concentration separately and is calculated using the equation shown on the slide. In the following slides, the acceptance criteria for the parameters described in the previous section and as recommended by the European Union Reference Laboratory for food and feed are described. The recommended acceptance criteria for the key assay performance parameters described in the previous section of the presentation are presented here and on the following slide. Firstly, the assay design must be specific to the target of interest. The primers and probe should lack secondary structure as well as the capacity to hybridize to themselves or one another. The assay PCR efficiency should be 100% plus or minus 10%, and the R-squared coefficient for the standard curve should be equal to or greater than 0.98 or 98%. The working range of the assay should span the values typically expected to be present in the sample analysed, and the limit of detection should be equal to or less than 25 copies of the target DNA, with an associated level of confidence of 95%. Continuing with the recommended acceptance criteria, the limit of quantification should be equal to or less than the lowest value at which the repeatability relative standard deviation is less than or equal to 25%. As a rough guide, the expected LOQ for a typical taxon-specific marker would be expected to be in the region of 50 target copies. An acceptable level of bias across the working range is plus or minus 25% of the reference value. And finally, in terms of the level of acceptable intra-laboratory variation for an assay, as determined by calculating the repeatability relative standard deviation across the working range, a value at or below 25% would be considered acceptable. Where an assay does not meet the validation requirements, it may not be fit for purpose, and the following remedial actions should be considered. Either attempt to re-optimize the assay in order to improve its performance and then repeat the validation process. Alternatively, redesign the assay or identify an alternative target and repeat the process of assay design, performance optimization and validation. Further information on the design of real-time PCR assays, as well as the optimization of quantitative PCR assay performance, may be found in the previous LGCE seminars indicated on the slide. In the following slides, the key points of the presentation will be briefly summarized, as well as the recommended acceptance criteria for the assay performance parameters described in the preceding slides. A list of useful tools, resources and references is also provided. To summarize this e-seminar, when establishing newly designed in-house quantitative assays or adopting those from the scientific literature or commercial sources, it is strongly recommended that their performance be validated. Assay validation generally involves the assessment of both assay specificity and assay performance. A list of key performance parameters that may be employed to evaluate assay specificity and assay performance are listed on the slide. Evaluation of assay specificity involves determining the specificity of the assay for the target sequence, as well as an evaluation of the structural conformation of the oligonucleotides. Evaluation of assay performance involves the determination of a number of assay-specific parameters, which include PCR efficiency, linearity of response, limits of detection and quantitation, as well as estimation of bias and precision. The values obtained may then be compared against published critical values for acceptance, such as those provided by the European Network of GMO Laboratories, in order to determine if an assay is fit for purpose. The acceptance criteria for the key assay performance parameters described in this e-seminar are summarized on the slide. A number of tools, useful resources and reference publications are freely available. Some examples of guidance documents relevant to the topics covered in this e-seminar are listed on the slide. On this slide, some examples of publications relevant to the topic are listed. In addition, a number of software applications that may be employed in the design and validation of real-time PCR assays are shown. We would like to acknowledge the support received in preparing, delivering and funding this e-seminar, in particular the Department for Environment, Food and Rural Affairs, the Government Chemist Programme for 2017 to 2020, the Department for Business, Energy and Industrial Strategy, the Food Standards Agency and Food Standards Scotland. And finally, we would like to thank you for your attention.